We are very excited to have Jason and Amanda here today to talk to us about their work looking at both the ecology and the ecosystem services of inactive hydrothermal vent deposits. And with that, I'm going to hand it over to them. Looks good, Jason. Just unmute yourself.
Are we having audio difficulties, Jason?
I was, thank you. Can you hear me now? I was like frantically looking for the mute button and not finding it.
Great.
'Cause
Over to you.
Sorry about that. Thank you. Thank you for the invite from COBRA. We're off to a great start. It's our pleasure to talk to you for the next half hour or so about the ecology and ecosystem services on inactive hydrothermal vent deposits. This is work that Amanda and I have been doing for the past 6 years. For most of it, we'll go back a little bit further in history. Before that, and just if you are not familiar with who I am, I'm a professor in the Department of Oceanography, Texas A&M University.
Hi, I'm Amanda. I'm an assistant professor at Louisiana State University.
Thank you. So, and I'll start off, give an introduction, and then Amanda will jump in later. So I wanted to start off with just a really broad background and remind everybody that hydrothermal vents are globally distributed ecosystems. You can see some photographs from different vent systems around the world. These are places where deep seawater is heated below the seafloor and comes back up out of the seafloor as very hot, greater than 400 degrees Celsius often. No oxygen and a lot of reduced metals in those hydrothermal fluids, as you see coming out of these vents here. And when that mixes with cold, deep oxygenated seawater, it precipitates the hydrothermal plumes that you can see in these images, and also precipitates the sulfide chimneys that you also see. So, and in this map, this is a slightly older version, but this shows that the majority of hydrothermal vents occur at plate boundaries where either new seafloor is being made at mid-ocean ridges or in subduction zones. On this map, all of the dots in red indicate locations where we have seen hydrothermal vents, either with manned submersibles or video or some other imagery. And in yellow are places where the hydrothermal vents were detected in the water column, usually looking for a plume for something indicative of vents such as elevated manganese or iron or methane that you don't typically find in seawater. So, and you could see there's in the southern parts of the world, not as many vents have been located, but likely the density of vents is similar there. So I point this out mostly just to remind us that hydrothermal vents are kind of globally distributed ecosystems. They're often thought of as places where kind of extreme chemistry and biology might occur, and to some extent that's the case, but I also like to remind us that they're quantitatively important components of the ocean system. And the kind of clearest way I can illustrate this is just by reminding us that as much hydrothermal fluid enters the deep ocean as river water enters surface oceans, and therefore we tend to think of rivers as being important. So hydrothermal vents, even just by volume of flux, must also be important. So not only are they interesting and fascinating, they're also just quantitatively important ecosystems. The hot fluids that are coming out of those chimneys on the left here, we have a picture of an active vent, and these have been originally discovered in the late 1970s and studied ever since then. And so these fluids support vibrant animal communities, often supporting organisms only found in hydrothermal vent environments, such as these tube worms that we see in the Eastern Pacific. But eventually, all vents will stop venting for a variety of reasons. And so on the left here, we see a picture of an inactive hydrothermal vent. And I put this out just to show that to remind us that the active vents are supported very much by these fluids that are coming out of the vents, and whereas anything that would be living on an inactive vent is going to be living off the rock itself because there's no longer fluids, and this is a colder ecosystem. And so coming back to this map, essentially everywhere that there is active vents, there is likely, if not now, eventually to also be inactive vents. And so as much as active hydrothermal vents are globally distributed systems, so are inactive ones. They are less well studied, at least from the biological standpoint. And so these stars indicate places where papers have been published with focusing on the biology of inactive hydrothermal vents. There's probably a few stars missing from papers in just more recent times, but you can see that it's a lot less than the sites that we've surveyed for active hydrothermal venting. So, and I will point out this star right here represents 9 degrees north on the East Pacific Rise, and this is the site that we're going to be talking about, where a lot of the data we'll be presenting it comes from. Okay, so why study inactive vents? So a few reasons. So I mentioned all vents stop venting at some point. And it turns out these structures are quite long-lived, so they can remain on the seafloor for tens of thousands of years based on studies that have dated the age of the structures. They're made of reduced and oxidized sulfides, iron oxides. These are all things that are fuel for microbial energy, right? And so essentially, they're long-lived microbial ecosystems. And so, Amanda and myself are microbial ecologists. And so the prospect of studying something that's kind of globally distributed and really good food for microbes is really fun. So we study microbial populations on these older inactive sulfides with the goal of characterizing microbial community and quantifying rates of metabolic activity that are relevant to ocean biogeochemistry, such as carbon fixation and nutrient utilization. So, and you can see in the bottom, this is a video of a site that we call Mush Pit, because it looks like it's giving a rock on sign if you look at the other side of the chimney, and also a nice picture of another inactive hydrothermal vent. The study of the microbiology of inactive hydrothermal vents doesn't go back that long. As I mentioned, 1977 was when we first discovered hydrothermal venting on the seafloor. And this paper by Suzuki and colleagues in 2004 is the first paper that was specifically looked at microbiology on inactive hydrothermal vents. So it's only been the last 2 decades, at least on the microbiology side, that people have really been looking at these ecosystems. And I just want to point out 2 things in this particular table. So in this study, Suzuki and colleagues collected samples from the Southwest Indian Ridge and also near Japan, some hydrothermal vents closer to Japan. And there's 2 things that they looked at that I want to point out. One is they did cell counts, looked at the population density of cells on these structures and found them to be 10 to the 7th, 10 to the 8th cells per gram of rock. If you're not used to thinking about things in that order, in that way, I will point out that this is about 4 to 5 orders of magnitude more cells in terms of density than you would find in the background seawater that these vents are sitting in. So they're really oases. And to further kind of hit that and make it make sense, a comparison would be, so both Amanda and I work at schools with very large football stadiums, Tiger Stadium at LSU and Kyle Field here at Texas A&M, and they both fit close to 100,000 people. And so the difference between what's on the vents and what's in background seawater would be like, background seawater is representative of 1 fan sitting in the stands cheering on the football team, and hydrothermal vents is representative of 100,000 fans sitting in the football stands and cheering on the football team. So there's a high population density on these structures. And the other thing that came out and has remained or been confirmed in subsequent studies is just that, on actively venting hydrothermal vents, there tends to be higher proportions of archaea. Sometimes, the majority, sometimes less, but they tend to be there. For whatever reason, on inactive vents, these are dominated by bacteria. So you see here that essentially this is quantifying bacteria to archaea as a ratio, and more than 95% of the population was bacteria. And this remains to be the case in follow-up studies. So going into what taxa are actually present in these vents, that's what some of the subsequent studies did, and I wanted to point out, so this is two figures. One is from a paper that I put out a little while ago, showing essentially a composite inactive vent chimney, the taxa that are present, and then from a variety of papers that had already been published, what we find on active vents. And so on the left here, we have a bunch of different colors representing the proportion of different bacterial taxa you might find on an inactive vent, and the same here for the what you might find on an active vent. And all I really want to point out here is that those colors are very, very different, right? And you see in particular on the active vents, we see a lot of what at the time were called epsilon proteobacteria. We now call them Campylobacteriales. And Aquificae, and these are bacteria that are thermophilic, so they need high temperatures. They also live off the fluids that are coming out of the vent. So it makes sense once the vent stops venting that you essentially lose these populations, and other organisms, in particular gamma and in blue and alpha in yellow, proteobacteria tends to take over and kind of grow in and do what they do, right? So this indicates an ecosystem succession happening on those chimneys. So, and I want to point out some work by other people too. So this is from a really nice paper by Shingo Kato and his colleagues, where they sampled microbial communities from active chimneys here highlighted in red, and inactive hydrothermal vents highlighted in blue, all from the same fields, and then compared statistically their microbial communities to each other. And what you could see that is that essentially we have one big branch where all of the active chimneys are showing that they are much more similar to each other than they are to any of the inactive chimneys, and vice versa. All of the inactive chimneys are more closely related to each other than to the active chimneys, kind of supporting this idea of an ecological succession taking place. So I'll bring us now to the field site where we did most of our work. And so we've been lucky enough, this is 9 degrees, 50 minutes north on the East Pacific Rise, which you can see here. This is Mexico and Central America. And the East Pacific Rise is this feature on the seafloor right here. 9 degrees north just refers to the latitude at which where we find this. This is, one of a few long-term kind of study sites that we have in the hydrothermal vent world. And so people have been studying this particular region of the seafloor for the past 30 years or so. And so we initially started going there just because it's a well-known location, and it fit what we needed for our initial experiments. We've been lucky enough to, since 2019, be able to go out there on 5 different cruises using either HOV, the submarine Alvin, or ROV Jason, and often in combination with the autonomous underwater vehicle Sentry. I saw Brian on the call. Thank you, Brian, for supporting us. And you could see that in this map of the system that most of these blue dots are active hydrothermal vent sites, and most of these, this kind of crease that you see here is what we call the axial summit trough, or essentially right where the 2 plates are coming apart. But I do want to point out that a lot of the, not all, but a lot of the inactive features are actually off axis, and so you'll see that in a minute. So I mentioned a little bit earlier in this presentation that one of the ways people look for new venting sites is by looking for chemical signatures in the seawater of active venting, and that works great. But that's a lot harder to do for inactive vents just because they're not actively venting, so there's much less of a signal. If you get really close, you can maybe detect something, but you almost have to bump into them. And so these have traditionally been found more or less by people that were already studying a site that, with a robot or a submarine and happened to notice inactive vents nearby. But in recent years, AUVs have been used more and more. And so I mentioned we have been using AUV Sentry to, in collaboration with collaborators, this map was made by Janai Wu, who's at Woods Hole now, to look for, to map the region, also to look for events. And in this map here, this is that same axial summit trough that you saw on the previous map. And all of these dots are structures that could be essentially hydrothermal vents. However, they need to still be checked. And so it turns out when you identify a lot of sites of interest, but then when you go look at them, most of these are actually not vents. A lot of them are things like basalt pillars. However, some of them are vents. And so some of the sites that we've discovered, including Lucky's Mound right here, which you can see It looks like this when you zoom in, and here's a side profile of it. We found specifically because we used an AUV to map the region. So through these cruises, we've sampled three kind of broadly defined types of samples, either actively venting chimneys, and so these are your kind of classic, black smokers, or depending on the temperature, but chimneys that were hot and actively venting when we collected them. Inactive vent deposits, which are chimneys that were clearly not venting. When you took a temperature, you poke it with a temperature probe, it is no warmer than background seawater. And then also we call them inactive, full stop, if they are, not venting and far away from any active venting, usually 100 meters or more. And that's in comparison to what we have called inactive but proximal chimneys, which are inactive vent deposits that, again, are not venting at the time of collection. And the temperature was not any different than ambient seawater, but maybe nearby active hydrothermal venting. And so an example would be this structure right here, which is a fallen down chimney. Don't know when it fell down, but it for sure was not venting when it was sampled. There was no fluids coming out of it. It was, background seawater. But you could see in the background, if you're a 9 North enthusiast, you might recognize the bottom of the Bio 9 chimney structure. And so this feature might be influenced by hydrothermal plume fallout. And so we wanted to be able to have some nuance, and so kind of categorize samples like that. And so if you look at just the taxonomy of what microbes reside on these structures, here we have an MDS plot, which shows you essentially each one of these points represents a different sample from which we extracted DNA, did a PCR survey to identify proportions of different archaea and bacteria in these samples, and then use some statistics to compare these samples to each other. And so essentially, the closer two samples are in this space, the more similar their microbial communities are. And what I would point out to you is that there's essentially a gradient based on activity. And so we could see on the left side of this figure, in red is all of the actively venting samples. On the kind of right side in blue, we see all of the inactive vents, seawater all the way kind of furthest to the right. And then these, what we were calling inactive but proximal samples, reside kind of in between and reflecting the fact that they are different from active, but still bear some resemblance to the inactive and active chimneys. And I also want to point out, so most of the data we're talking about is from the East Pacific Rise, but this pattern in particular of the inactive vents looking very different from the active vents holds up when you compare them to other deep sea environments as well. And so from this nice paper from Han and colleagues that came out a few years ago, they took the samples available at the time of inactive vent microbial communities, the taxa present from the Indian Ridge and the Atlantic Ridge and the Pacific Rise, so different places in the ocean, and they compared them to other marine settings, including active vents, water column, hydrothermal fluids, and found them to be unique, or occupying their own space in these statistical analyses. These are two different ways of looking at this same samples. And you can see in blue we have inactive chimneys, and in red all other types of samples. And so what this tells us is that the communities specifically on inactive hydrothermal vents and microbial communities are unique, and therefore, if you take the next step, likely to provide unique ecological services. Some examples could be iron oxidation, sulfide weathering, and primary productivity. So I'll hand it over to Amanda to take it from here.
Thank you. So through some of those studies that Jason mentioned, as well as others, we see a lot of evidence that the microorganisms that we find on inactive deposits have the genomic potential to fix inorganic carbon. But we wanted to see how that actually translates to rates of primary productivity in these environments. So we measured the incorporation of C14 labeled bicarbonate into cellular biomass by vent-associated microbial communities along the East Pacific Rise. So we did these measurements on active vents, inactive vents, and that inactive but proximal sort of setting that Jason already described. And we did these measurements over the course of about 5 or so years and on a dozen or so different sites along the EPR 9 North area. And I want to point out that these measurements were taken and pertain to the rock-associated microbial communities, so they are not necessarily reflective of microorganisms and activities that might be found in hydrothermal fluids. So these results that we garnered over these last 5 years or so are summarized here in this figure, with the rates expressed as nanomoles of carbon fixed per gram of rock per day. And while there's a fair amount of variability between these samples and sample types, for example, we can go to the same vent edifice and sample at 2 different places, and we can get pretty different carbon fixation rates. But that variability means that there isn't a lot of statistical difference between these 3 vent types. So these results suggest that the microbial communities that we're finding on inactive vents are capable of fixing inorganic carbon and contributing as much to local primary productivity as some of their more active counterparts. So in collaboration with Ann Dekas at Stanford, we dug a little deeper and were able to examine the uptake of 13C bicarbonate at a single cell level using nanosims. An example of those results is shown in these 2 images here, with cells that took up the substrate appearing in more of a reddish color. And so what we found was that actually only a very small proportion of the overall cell population was metabolically active at all. So for example, only about 16% of total cells were metabolically active on active vents and only about 6% on inactive vents. However, of those active cells, a large proportion were carrying out primary productivity and fixing inorganic carbon. So about half of those active cells on active vents were fixing inorganic carbon, and about a third of the cells on inactive vents were doing that. So these methods further demonstrate that carbon fixation is occurring and is prevalent in inactive vent systems. So next, we performed a metagenomic analysis of some of the inactive vent samples that we were studying for primary productivity to better understand the carbon fixation pathways being utilized and who the potential microbial players were. And so we found that the CBB cycle was the most prevalently used across our samples, but there was secondary contributions from the RTCA cycle and Wood-Lindahl pathways. So those genes associated with the CBB cycle were found to primarily belong to members of the gamma and alpha proteobacteria. And the Campylobacteria, Nitrospira, and the UBA 7883 group contributed more to genes involved in the RTCA cycle. And I want to highlight that the presence of organisms using the CBB and RTCA cycles was also supported by paired analysis of total organic carbon and microbial lipid isotopic values measured by Florence Schubatz and her group at the University of Bremen. So I always like to try and, frame my findings against what others have found. And that's what I'm showing here in this graph on the left. So highlighted in red are the carbon fixation rate ranges that we measured for active and inactive vents. And it's compared or graphed alongside other published rates that we found in literature for vent environments. So if the rates came from the vent rock or deposit itself, it's shown in black, and fluids are shown in gray. So we were the first to report measurements of carbon fixation rates on inactive vents. There was no comparison for that data, but we did find that our rates for active vents fell well within the ranges that had been reported in other studies. So our measurements track well with what has been reported in literature. And if we want to go a step further and take these rates that we measured at each specific rise and extrapolate them to the global estimate for C4 massive sulfide deposit volumes, we come up with a number that is close to the estimate that has been put forth for the amount of fixed carbon fueled by global diffuse flow venting systems. So there's, of course, a lot of caveats when making those sorts of extrapolations, but what our work was able to demonstrate is that inactive vents do host unique microbial communities, and that those communities are contributing to deep sea primary productivity on a scale that is perhaps comparable to their active counterparts. And so this really signifies that inactive vents could be a source of new organic carbon production on the seafloor that we typically have not considered.
So, and just briefly, but one of the other kind of ecosystem services that we've considered in our studies of the inactive vents are exoenzymes. And so exoenzymes broadly are a suite of enzymes that microbes will use to help them digest various aspects of organic carbon that they maybe can't bring into the cell kind of as a whole piece. And so they use these enzymes to break them down into smaller pieces. That's kind of the extent of what I think is needed to know to interpret this. What I wanted to point out is that one, particularly well-studied enzyme is alkaline phosphatase. In the kind of range of rates that we measured on EPR sulfides, the active ones in red, the inactive in blue, when that is compared to alkaline phosphatase activities from other environments, shown here on this figure, just the white boxes are showing the ranges from different environments. What we can see is that that range falls into kind of the upper range that we see for what tends to be measured in seawater, and kind of well within the range of what we find in marine sediments, which are considered an environment where these enzymes are pretty active. And so the way that I like to look at this is just, this is, an indication of biogeochemical cycling in these systems, and the rates at which this is happening, kind of like for primary productivity, seem to be fairly on the high end in comparison to other marine ecosystems.
Yeah, so our investigations into inactive vent microbial communities and activity led Jason and I and our collaborators to wonder if inactive vents represented a unique ecosystem in which we have chemoautotrophic microorganisms fixing inorganic carbon, and if that could potentially support higher trophic levels or animal populations. So here I'm showing you an image of one of our inactive vent sites, Sentry Spire, so named from the AUV Sentry that helped us find it. And I can imagine you looking at this picture and wondering, like, why are we spending so much effort looking at inactive vent deposits as from like an ecosystem perspective, when really it just looks like a pile of rocks? But if you focus on one little area and you zoom in and you zoom in a little more, all those tiny little white dots are actually animals. So this final image that popped up represents about a 10-centimeter wide field of view. So these animals are very small, and it probably isn't hard for you to realize why they were overlooked so long. And so it was very interesting to find these little tiny animal populations on sites that we looked at, and we have actually found them on every single inactive vent that we have explored so far. So this is a video from Lucky's Mound, which is an inactive deposit that's several hundred meters away from Sentry Spire. And I just want you to highlight the density of all these little white dots. So we can find several of them in like a small cubic centimeter. So while they are small, they are actually quite abundant. This is a photo compilation of some of the animals that we have found living on or in close association with active vents through work done by our collaborators, Lauren Molyneux and her group at WHOI. And some of these animals, like the isopods and amphipods, are mobile, and they probably are not living exclusively on inactive vents. But these gastropod species, these snails and limpets, we start to wonder about. So highlighted in yellow are two animals, Melodramia gallerone and Sutilla zona. So far, we have only found these on inactive deposits that eat specific rise. This little guy highlighted in red is Neolepitopsis. And Molyneux and her group have found the larval stage of this limpet for many years around the specific rise, but it wasn't until we started looking at inactive deposits that we actually found the adults of the species. And finally, these two, Melodramia, laurelin, and Tilperion, were new species that were found at these specific rise. So there are two new species to science that was found through the work of our collaborators. So I only highlighted a handful of the animals that we find at inactive events along these specific rise, but they are not insignificant members of the animal community. And that's demonstrated through this relative abundance figure. So these handful of species, shown primarily in the bluer tones, comprise the bulk of the animals that we have observed thus far. So a few species seem to really dominate and thrive on inactive event ecosystems, at least at the sites we've looked at so far. And what is especially important to highlight, I think, is the fact that these animals are not simply congregating or just finding any hard seafloor substrate to land on and to sit on. This NMDS plot demonstrates that the animal communities that we're finding on inactive vent deposits are distinct from the animal communities that we observe in peripheral basalt surfaces. So there is something unique about the inactive vent deposit itself that appears to make it a viable habitat for this select unique group of animals. And so it is perhaps possible that some of these animal populations are actually endemic to inactive vents. So one of the things that may make inactive vents appealing to certain animals is the microbial communities. So we're just beginning to dig into this sort of connection between inactive vent microbes and animals. But when we compare the shared microbial species between what we find on the inactive deposit rock itself versus what we find in background seawater, and compare that to the microbial communities that we find on the whole animals, or specifically in their guts, we find that there are many more microbial species shared between the vent deposit and the animal, especially their gut, than the water column. So this may suggest that there's some connection between the animal and the vent microbial community. Perhaps the microbial community on the vent is serving as a direct food source for these animals. And to further highlight this potential, here we're showing the degree to which microbial communities from the guts of two different animals are shared with the microbial communities found on a few different environmental surfaces. So here I have inactive vent deposits, inactive vent deposits, basalt rocks, and background seawater. So now Neolepidopsis, I already mentioned, the adults have only been observed on inactive deposits so far at EPR, but Lepidodrillus is a little different. It typically has exhibited a much wider habitat range, and so we can find it on active and inactive vents. It doesn't appear endemic to inactive sites. And these habitat preferences between these two different species seem to be somewhat reflected in their gut microbial communities, such that the microbes found in the gut of Neolepidopsis are most similar to what we see on inactive deposits, while Lepidodrillus showed more similarity to what we find on active vent deposits, but it also shared a lot in common with inactive deposits and basalt rocks. And I want to point out that these specimens of both specimens, Lepidodrillus in particular, were exclusively harvested or collected from inactive deposits. So we're still in the early days of this, as I mentioned, but we're starting to find connections between the inactive vents, the microbial communities, and the animal communities. And so we're working to gain a deeper understanding of what the inactive vent ecosystem looks like and how it's functioning.
So we'd like to end with just a few slides thinking in kind of going back out to the broad context. And so this is a map, again, of where hydrothermal vents are located in the world. This was made by Ross Parnell-Turner at Scripps. But importantly, I want to draw your eye to the areas that are highlighted in yellow. And so these are regions where the mid-ocean ridge has been licensed for seafloor massive sulfide, which is what SMS stands for, exploration areas. And so these are typically licenses that are requested by countries. And the reason that this has been done is because hydrothermal deposits contain economically important metals. So as we saw earlier, right, active hydrothermal vents are host to large, charismatic, and unique animal communities. I think the microbes are also charismatic. And also they're venting very hot, usually acidic fluids. And so they're kind of the combination of a harsh environment to try to harvest, as well as the already known and well-documented unique animal communities, have kind of lowered those on the list of kind of interested targets in regards to mining interests. However, inactive vents have been discussed for some time as good targets because largely they have the same minerals that the active vents have, and also it was thought that they didn't have very much biology or anything that might cause need management. And so I'll close by pointing to this paper that came out a few years ago. It was led by Cindy Lee Vandover and came out of a workshop by people both from the science community as well as government and mining interests, which was focused on specifically inactive sulfides and the types of information that would be needed to consider potentially mining them. And the three takeaways that we came up with were that what's needed is to undertake fundamental ecological descriptions and study of ecosystem functions and services associated with hydrothermally inactive sulfide deposits. 2, to evaluate potential environmental risks to ecosystems of inactive deposits through research. And 3, to identify environmental management needs that may enable mining of inactive deposits, noting that mining of some of these deposits may have reduced environmental risk compared to other seabed mining activities, but this must be validated through scientific research on a case-by-case basis. And what I would say is I think that the work that Amanda and myself presented and of our colleagues in this field has really addressed number 1 the most. I think that number 2 and number 3 are unconstrained, and I think that some of the work in particular finding potentially endemic animals on these structures force us to kind of rethink what management might look like, and the fact that these actually are potentially unique ecosystems. And so with that, I'll summarize briefly. So we showed you today that microbial communities on inactive hydrothermal vents are statistically different from those on active vent deposits, that there is active carbon fixation on inactive vent deposits, and it appears to be similar in rate to active deposits and diffuse hydrothermal flow. There are potentially endemic animal communities on inactive vents. For sure, there are unique communities not identified at active sites, and they are likely eating microorganisms on the inactive sulfides. This all has, I think, should be at least part of the discussion, thinking about potentially mining of these sites. And there's just so much more science to come. So I will leave you with, thank you for listening, leave you with a pretty picture from the Juan de Fuca Ridge, and also the kind of obligatory photos from the various cruises that collected these samples. And in particular, we want to acknowledge our funding from the National Science Foundation, which funded all of the field work and most of the research, as well as funding from the Simons Foundation. So with that, I will thank you, and we can take questions.
Wonderful. Thank you.